Hello students, welcome to Misostudy again. In today's session, we will be discussing about certain questions, CBSE pattern question exercise from the chapter Sexual Reproduction in Plant. We have discussed the whole chapter, now it's time to revise certain questions. So, I have divided the question into different categories, some questions from 1 mark, some 2 marks, some 3 marks and some 5 marks. I will tell you general strategies, how to proceed in that question, what you have to answer. I will not give you a complete explanation of the answer because that is already mentioned in the sessions. So, you can refer to those sessions, but I will tell you how to proceed in such type of questions. Now, let us start. The question number 1, 2, 3 are of 1 mark. Question number 1 indicate angiosperm bearing unisexual flowers are said to be either monoecious or the dioecious, explained with the help of certain examples. Now listen very carefully. Here it is written over here, the monoecious. Now what do you mean by the monoecious? That means both the sexes are present within the same flower. Both the sexes are present. Whereas in dioecious, both the sexes in separate plant. Both the sexes are present in separate plants. Right? That means, like what do you mean by this? That means the pistillate, like your pistillate, the staminate, both are present in the same plant in this case. In this case, they are present in the separate plant. Now, they are asking one example as well. In this case, you can take an example of a diamase, and in this case, you can take one example of a papaya. Right? So, this was the question number 1. This is of 1 marks and this is how you have to answer. Let us move on to the next question, question number 2. How do pollen grains of Velisneria protect themselves? As you all know, the pollen grain of Velisneria, they uses water for the distribution of their pollens. So, they uses water. So, they have one covering which we call it as mucilaginous sheath. They have one mucilaginous sheath present, right, which protects itself, which protect these pollen from the harsh action, right. This is how they protect themselves. Now, let us look at the question number 3. This is also of 1 marks. Mention the function of coleorhiza. Now, what is a coleorhiza? This is a protective covering which is present on the grasses, on the root of embryo in grasses. Protective covering on roots, or which roots? Embryo roots of grasses. Right? So, this was the question number 3, the first category, 1 marks. So, so note down this, then we will proceed towards the next category. So, students, hope you have noted down. Now, let us move on to the next category. This is question number 4 and you have to answer in like these are the 2 marks questions. So, you have to answer accordingly. Question indicates a mature embryo sac in a flowering plant may possess 7 cells but 8 nuclei. Explain with the help of a diagram only. Now, for that I need, because I have to draw a diagram, for that I need some colored chalks. So, this is that diagram which I have already drawn in the sessions, but this is just a rough diagram which I am drawing right now. So, they are asking an embryo sac. So, in case of an embryo sac, this is an embryo sac, the 2 ends are there. This end is called as a chalazal end. This end is called as a micropilar end. Right? In the micropilar end, these are the synergids present. Synergids. right. Here the 3 antipodal cells are present. Antipodal cell. In the center, you can see a polar nuclei is present. Polar nuclei. Now, count the number of cells. Here you know one more atom is present, right? So, just count the number of cells which is present in it. This is number 1, this is 2, this is 3, this is 4, 5, 6 and 7, right? And the same thing question is asking. So, in this case, you have to draw this diagram and you have to show that the 7 cells are present. Now, another part of the question indicates that in this case, the 8 nuclei are present. Yes, it's quite, it's like 1, 2, 3, 4, 5, 6, 7, 8. So, these are the 8 nuclei which is present in it. This is how you have to explain. With the help of a diagram, a neat and clean diagram you should draw, and you have to explain accordingly. Now, let's move on to the next question, question number 5. A pollen grain in angiosperm at the time of a dehiscence form an anther, could be 2-celled or 3-celled. Explain how are the cells placed within the pollen grains when shed at the 2-cell stage. Now, they are asking when we talk about these pollen grains. They are asking about these pollen grains and they are asking about the anther, right? These anther, they are in development stage. Developing stage, right? They are present in developing stage, and in this developing stage, they divide unequally. Divide unequally and produce 2 types of cell. 2 types of cells. The first cell is called as a vegetative cell. Vegetative cell. And second cell is called as a generative cell. Generative cell. Right? In 40% of angiosperm, 40% of angiosperm, it is seen that 3N condition is seen in case of anther. Whereas in 60%, in majority of cases, the condition is 2N only. That means the 2 type of cells, vegetative cells and the generative cells is present. Now, what happens in case of these 40% where the 3Ns are produced? In this case, generative cell, they further divide into the 2 male gamete. Male gamete 1 and male gamete 2. So, hope you are getting what I am trying to say, that in 40% of the angiosperm, the 3N condition is there. That means 1 male gamete, you can count it as a 1, 1N, 2N and the 3N, right? 3N condition is there. Whereas in 60% of the angiosperm, only the generative cells and the vegetative cells are present. This is what you have to explain, and we have already studied the same in the sessions. So, as it is just a 2 marks question, you don't have to explain like a huge, or you don't have to give a huge explanation. Just give a point-wise explanation. What is important, just give that explanation only. No need to write whole stories. Now, let's move on to the next question, question number 5. Now, another important question, write the difference between the tender coconut water and the thick white kernel of a mature coconut and their ploidy. First of all, tell me one thing, they are asking about the ploidy. They are asking about the coconut water ploidy and the white kernel. That means they are asking about the endosperm. In this case, the ploidy is always a triploid. They are always a triploid. Now, to talk about the tender coconut, and this is, they are asking about a white kernel. White kernel. in case of the tender coconut, you can see the water is there, the endosperm is there. In case of the water, a watery condition is there, and this tender coconut, the endosperm develop from nuclear endosperm. In this case, it developed from cellular endosperm. Cellular endosperm. This is the basic difference between a white kernel and the tender coconut water, right? So, a very short, and you have to answer this in a brief. 1, 2, 3, 3 to 4 line, that's it. It is enough for your 2 marks question, and this is how you have to answer, right? Let's move on to the next question. Again, this is of a new category. Describe the structure of a 3-celled pollen grains. Hope you remember the diagram. I'll be giving you a rough diagram of a 3-celled pollen grain. So, if you remember, in the session I have drawn one diagram of this 3-celled pollen grain, right? Here the exine is present, so this is called as a tube. Which tube? Pollen tube, right? Here this will be called as a tube nucleus. Over here you can see the 2 cells are present. They will be called as a male gamete. Male gamete, right? Surrounding this male gamete, can you see a sheath is present? This a red color, which I am trying to show, a red color sheath is present, which we call it as a cytoplasmic sheath. Cytoplasmic sheath is present, right? This is a whole structure of a 3-celled pollen grain, and you have to explain about this, as we have already done the same in the sessions, right? The diagram should be neat and clean. You should draw the diagram with the pencils only. No need to use any other color. Just use only pencil. Neat and clean diagram, are well labeled, and try to label all these labeling on one side. Do not spread all these labeling towards every side. So, just do that. Now, this was the question number 7. Let's move on to the next question, question number 8. Differentiate between a perisperm and endosperm, giving one example of each. So, perisperm, endosperm. First of all, the perisperm. How perisperms are formed? They are formed from persistent remain of nucleus. Now, how these endosperms are formed? These are formed from primary endosperm nucleus. Primary endosperm nucleus, right? Now, perisperm is a part of seed. It is a part of seed. Whereas these endosperms, they act as a storage of food material. Right? When you look at this perisperm, they are in a dry condition. Quite hard they are. They are present in a dry condition and they are fleshy or they are having a watery fluid present. Watery. Right? In case of the perisperm, we have one example like black pepper. Black pepper. Endosperm, you can take an example of a coconut, coconut water, right? Coconut water. So these are the certain, I have just mentioned a few differences. You can add on a few because we have already completed the same in the session. You have to add on few because if I try to explain each and every difference in the today's session, the session will be very much long. So, my main motive is to tell you what you have to write in this question and how you have to proceed. So, these are the very easy questions. Now, let's move on to the next category. So, these are the 5 marks question. Draw a diagram of an enlarged view of TS of one microsporangium of an angiosperm. Label the tapetum, these middle layers, endothecium and the microspore mother cell. Now, hope you remember the diagram. That was a very easy diagram like this, right? You have to draw this one diagram as well as another diagram which I have drawn. You can make a cut like this, right? And towards the outer side, one layer is present which is called as a epidermis. This is called as a epidermis, right? After that, another layer which is present is called as a endothecium. Which layer? Endothecium. I am just giving you a rough diagram. After that, 2 to 3 layers of another cells are present, which we call it as a middle layer. They will be called as a middle layer. Now, after that, one very important layer is present, which we call it as a tapetum, which provide nutrition. This layer is called as a tapetum, right? Towards the inner side, these microspore mother cells are present. Microspore mother cell are present. Now come back to question. Draw a label diagram and an enlarged view. Enlarged view. You don't have to draw this diagram. This is not an enlarged view. So in the rough, you can draw this diagram and later on you can explain the detailed diagram which was regarding this. This is not a complete diagram, but you have to explain like this. A very well labeled, a beautiful diagram you have to draw if such type of question comes in your exam. So you have to label the tapetum, middle layer, endothecium and the microspore mother cells. All of these 4 parts I have already labeled. Now, mention the characteristic feature in the function of the tapetum. You all know the function of the tapetum, that it provide nutrition, right? The main function that it provide a nourishment. So third is explain the following giving reason pollen grains are well preserved as fossils. Now, third part, as you all know that pollen grains, the outermost covering of the pollen grain, they have exine present, right? And which is a protective layer, and this exine which is present is made up of sporopollenin. Sporopollenin, irrespective of action of alkali, irrespective of action of any kind of acid, nothing happens to these pollen grains. So they can be easily preserved in the fossils. Let's look at the B option. Pollen tablets are used of the public these days. The reason is that they are a very good source of the nutrient. That is why they are used by many people. So this is how you have to answer. Like, it is quite definite that when you will be appearing for the exam, a long answer type, if such long answer type question will be asked, in that case the question will be made up of different parts, and each part will be having a specific mark. So you have to answer accordingly. Try to make a diagram, because diagram makes your whole answer very much it is very much impressive, right? Now let's move on to the next question, question number 10. So explain the event after pollination. Now the pollination has happened. We have to explain the exact process. For the formation of a seed in angiosperm, mention the ploidy level. Now, I will not be explaining the whole process in detail, but in this case, what you have to explain is first how the double fertilization happens. Double fertilization. Hope you remember the session I have discussed regarding the double fertilization. Very, very, very important step. Second, you have to explain the another, that is the embryo formation. Embryo formation. You also have to explain regarding the endosperm formation. All these processes you have to explain with neat and clean diagrams. With neat and clean diagrams, right? Let's look at the second part. Mention the ploidy levels of the cells of the different part of albuminous seed. First of all, in this case of embryo, embryo will be diploid, whereas the endosperm, that will be triploid. That will be triploid, right? So students, we have discussed 10 questions from a different category. Each of questions are from different category. This is how you have to explain. If you have any doubt regarding what to explain in this, how to explain in this, how to explain to, that I have already explained to you. What to explain, you can go back to the session. If you remember the session, that is well and good. If you don't remember, that is very bad at your part, because if you have seen the session and still you are not able to answer these easy questions, that is very bad part. So go back to the session, watch them again, draw the diagram, make your own notes, and then explain these questions by your own. And also you can practice certain questions which are mentioned at the end of each chapter, at the end of each chapter which is mentioned in the ebook. Do practice them. So we'll meet you in next session. We'll discuss a new chapter. Till then, take care of yourself, and thank you so much students for watching this.